Welcome, my name is Sheila Malloy and I'll be co-supervisor on this MRC-led PhD project entitled The Epidemiology of Antifungal Resistant Candida in Southern Africa. I'm a senior lecturer in epidemiology based at City St George's in the UK, and I also hold an honorary position at Wits University in South Africa. Before introducing the project and the supervisory team, I'd like to give you some background to the research and highlight the importance of this field. So what is Candida? Candida is a type of fungus that colonizes the skin and gut, but can also cause severe fungal sepsis, with around 1.5 million cases of invasive candidiasis occurring per year, mostly in critically ill or immune suppressed patients. There's a very high mortality associated with invasive candidiasis, with around a third of deaths attributable to Candida infection. Treatment options are diminishing due to rising levels of resistance to the limited armamentarium of antifungal drugs. Antifungal resistant Candida are deemed an urgent and serious threat to global health by the CDC, with the WHO also listing 5 Candida species as urgent and critical priority fungal pathogens. There are only 3 classes of antifungals used for the treatment of candidiasis. The use of amphotericin B is limited by cost and toxicity. Widespread use of azoles has driven a global shift towards intrinsically resistant species, as well as increasing levels of secondary resistance. An increasing reliance on echinocandins, which are now the first-line treatment of invasive candidiasis, is really a ticking time bomb for the final useful drug class. In addition, there has been an emergence of often multi-drug resistant species of Candida glabrata and Candida auris, and global spread of a new azole-resistant strain of Candida propsilosis over the last few years. This map shows the global spread of Candida auris, you can see here in red, and where auris and azole-resistant C. paropsiliosis are both prevalent, is shown here in orange. And really you'll see that there's no epidemiological data available for the large majority of African countries. In South Africa, however, robust epidemiological data, driven largely by Nilesh Govinder's group based at NICD, has documented this shift away from predominantly azole-susceptible species of Candida albicans, shown here on the graph in blue, towards propsiliosis and auris, shown in yellow and red. These are now the dominant species, with most exhibiting azole resistance. And the map here shows the clustering of Candida auris cases around the Gauteng region, where Johannesburg is located, but more recent data shows that auris is now endemic throughout the country. In light of the epidemic of antifungal resistant Candida in South Africa, the lack of epidemiological data from neighbouring southern African countries really is concerning. However, Prof. Govinder's group have now expanded their surveillance to conduct hospital colonisation surveys and wastewater sampling in several southern African countries, detecting Candida auris for the first time in Botswana, Mozambique, and in wastewater samples in Zimbabwe. Several studies have shown that antifungal exposure is one of the main drivers of the emergence of antifungal resistance in Candida, causing infections of colonizing patients. However, most studies are conducted in settings with relatively low burdens of antifungal resistance. This PhD project will be centered around the first study of antifungal resistance emergence in high burden settings within Southern Africa. So the PhD, which is entitled The Epidemiology of Antifungal Resistant Candida in Southern Africa, aims to firstly describe the epidemiology of Candida colonization or infection in approximately 1,000 patients, as well as environmental contamination on ICUs in South Africa and Mozambique. Also aim to describe the antifungal prescribing practices using WHO point prevalence surveys at these sites, and investigate using bioinformatic genetic linkage and modelling techniques the dynamics of antifungal resistance emergence, acquisition and transmission in relation to antimicrobial exposure and infection prevention and control interventions. This study will take place next year in 5 hospitals in Johannesburg and 1 hospital in Maputo, Mozambique. We aim to enrol about 1,000 participants admitted to adult ICUs at these sites and study the emergence of antifungal resistance in infecting isolates of Candida and isolates grown from serial swabs taken from the mouth and skin during the first 30 days of admission. The antifungal susceptibility will be measured and analysed in the context of antifungal drug exposure, IPC interventions and transmission from the environment. Isolates that demonstrate emergence of antifungal resistance will be sequenced to identify likely transmission routes and resistance-associated mutations. This PhD is embedded into a larger research network and programme of work entitled Combat Candida. This is led by Professor Nilesh Govender and Dr. Rachel Wake and includes researchers from across a number of institutes in the UK, South Africa and Mozambique. So in terms of your supervisory team, this will consist of a subset of the larger Combat Candida team. We are three co-supervisors, myself, Professor Harrison, both based at the Institute for Infection and Immunity at City St George's, and Prof Jarvis based at London School of Hygiene and Tropical Medicine. You'll also be supported by key collaborators from the Combat Candida project, Nilesh Govender, Rachel Wake and Tihana Beachnish. So our team has an excellent track record in research and impact, and importantly in ongoing career support and mentorship for our early career researchers. Prof Harrison leads a programme of clinical and laboratory research focused on driving advances in the diagnosis, prevention and management of invasive mycosis, including HIV-associated cryptococcal meningitis and Candida bloodstream infections. He's a seasoned research degree supervisor and mentor, passionate about ensuring impact, growth and sustainability of research. I am a senior lecturer in epidemiology and I've worked in the area of infectious disease epidemiology in low resource settings in Africa for almost 20 years. I have significant experience in the design, setup, conduct and analysis of clinical trials, implementation research and cohort studies in these settings, and I hold an MSc in medical statistics. While Prof Jarvis, he's a clinician, clinical scientist based at LSHTM, and he has specialist expertise in the delivery of trials and research studies, especially in inpatient settings in sub-Saharan Africa, also in clinical epidemiology and clinical mycology. As a team, we will train the PhD candidate in analytical skills, support the statistical modelling work and assist in translation of findings to impact in addressing this neglected area of global health research. Our three collaborators are clinical academics and internationally recognised experts in their field. Nilesh Govender and Rachel Wake are both based at Wits University in Johannesburg, South Africa, and as mentioned, they co-lead the Combat Candida programme of work, among a number of other projects, and Prof Beachnish leads translational research in the field of invasive fungal infections based at City St George's. So I hope you'll agree that this project aims to address research questions of global health importance, and hence is an excellent choice for an aspiring and ambitious researcher to make a true impact in this field of research. Thank you.